Right. Looks like we've got some people here. Got Dick and Morgan and Jesse. Hello.
Hello.
Hi.
Good morning.
Morning. So, any updates before we get into it?
Make the NetPyNE meeting this week conflicted, but I did bring up an email with them the Combined Harmony meeting and was asking them about how much overlap there was, or how much interest they had in that kind of at what point does computational neuroscience merge with computational systems biology?
Oh yeah.
And as well as like being really interested in terms of their just their overall like long range plans. And I got a little response back from one of the grad students there. I'll try and dig that up, but not much. I mean, basically, like that sounds cool, but I still think that like looking at the workshop that's this summer that James Glazer's putting together.
Yeah, the CompuCell 3D.
Yeah. And I forget if they bill it as. I think they're also doing a meeting at that time.
Okay.
Or something. It seems to be a couple events back to back. And that might be the excuse me. That might be the meeting to go to talk more about that. But just related, one of the things that they brought up is that it sounds like you might be interested in the kind of thing that the Human Neocortical Neurosolver group is doing at Brown, and that was funny because I was just was absolutely just talking about them, and they're gonna do an event like May 2nd, I think. And yeah, I'll probably post some of this stuff in like brain organoids.
Yeah.
Like for lack of and anyway, the only other thing is I did drop a link in DevoWorm orthogonal. Just somebody had posted this what I would call an ode to developmental biology on Hacker News.
Oh wow. Okay.
Aren't embryos amazing?
Yeah.
Like yes they are.
Yeah.
So, and you might I'd love to pick your brain a bit about INCF contacts and things at some point but this is kind of NarTechX related, but
yeah.
We're I think I told you that we got some board changes and things like that. But no, there was something else. Oh yeah, I met with a developer from Metacell.
Okay.
And Metacell do kind of contract software engineering for a lot of scientific projects, right? I mean that's kind of their thing, and so it was interesting because it was like, well, this is the people behind like the NetPyNE GUI. These are the people behind Virtual Fly Brain. The people behind and so that was also just a cool contact of them.
Yeah. Who was that? Do you know the name or?
Gopal. One sec. Gopal Krishna.
Okay. Yeah. I just asked because there are a lot of OpenWorm people in Metacell. It's Steve Larson is the CEO, and Steve Larson, of course, he's the sort of one of the people in OpenWorm who sort of started things. You saw in the talk that I gave a couple weeks ago. Yeah.
The the EI talk?
Yeah, I think so. Well, I mean,
Or no?
Steve Larson, he's done a lot of open room talks. If you go on YouTube or
Gotcha.
whatever, and you'll
Yeah.
see him. So that
Yes.
that's but
Okay.
so he's also at Metacell, and there are a number of people, senior contributors from OpenWorm, who are also sort of Metacell adjacent or Metacell involved. So
Gotcha.
yeah.
Gotcha. Okay. Well, it's in trying. I don't know if this. Sorry, this might not be DevoWorm relevant, but in trying to explain, the difficulty with brain-to-brain communication. I was trying to get at the fact that there were probably multiple languages of different brain areas, especially like different modalities, and basically trying to use the embryo and the kind of the collective intelligence that is required for kind of spatial morphogenesis as like obviously what at least is a good analogy for what's probably happening in the brain as well, and again, no one cell is in charge.
Yeah.
It's they all develop together, and they're all changing and learning each other's languages and things like that. Anyway, just a great ode to the wonders of developmental biology.
Yeah. Well, that's great. Yeah, thanks, Morgan. That's great. I'll actually talk about combine harmony in a little bit because I was looking at the materials and I thought it was kind of a nice conference. I don't know.
I'd seen, it was great that she joined because I had seen it months ago.
Yeah.
And as an aspirational thing, and because of course it was free, I signed up. I've got kids, I got responsibilities. I can't jet off to London.
Yeah.
But I did want to go. I mean, it looked like such a great agenda. It had such a great agenda. And, things happening. Yeah. Next time, or hopefully, they'll be in the States or something.
I'm joining the CompuCell 3D conference.
Oh yeah.
So
this summer, it's like
this summer,
late July, early August.
July, August. Yeah. I decided I should do that.
That's awesome. Awesome. I'm gonna try if I'm not shuttling kids places. There's super close to that is computational cognitive neuroscience.
Oh yeah, the conference.
That's like 6th to the 9th or something like that. Anyway, that's awesome though, because if I can't make it, I'd love to hear how that. His all his coursework is online. All his talks are online. They're such a great multidisciplinary mix.
Yeah.
Oh. Hi, I'm being bugged by a dog.
Okay.
You can hear her.
That's exactly
what she wants.
Yeah.
So you need brain-to-brain communication with the dog.
Yeah.
So I know what she wants.
So,
anyway, yeah.
Oh, Susan.
I got my tensegrity model to work using what we call a T4 configuration.
Oh, yeah.
Studied any tensegrity. But anyway, I got it to work, so now I can get some data, and that's all I need really. So
okay.
Nice.
What was special about the T4 configuration?
It shouldn't be stable, but it is.
Okay.
The compression elements come together in a V.
All right.
So it's like stacking one going this way and one going the other way. And you're putting them together, and I think they just push on each other the right way so they stabilize. I guess I should analyze that, but
Yeah.
if I don't have to, I won't.
Yeah.
Yes. They keep telling me, well, you have to make a diagram and show the forces, and for your thesis, we want to see this. And I'm going, oh, great. Just what I wanted to do.
Yeah.
Anyway.
Well, that's great.
So
yeah.
I got the hexagon model to work.
Good.
With there's 3 hexagons I've joined together in 4 places with a trijunction, and the trijunction is also a T4 configuration, and that works.
Yeah.
It's stable enough I can get data. So.
Yeah. So when you model it, what are you using to model it? What software?
I'm just using CompuCell.
Okay. And so you're getting like numbers out of it, like.
Yeah, I changed the strain on the members, and I can get a stress strain curve from it. That's all I need to do. I'd like to do some frequency analysis with it, but and then they said, well, you have to relate it to cells, and I'm going, well, cells are not a T4 configuration.
Yeah.
Not really. They do other things, including actively rearrange their parts. So yeah, I'm not sure what I'm going to say about that, but at least I can say that this simple stable tensegrity can be used to model that cell configuration. So yeah. So it's nice that that's working.
Well, that's great.
Because then I can write it out.
Yeah.
See, this works. Yeah. I'm done. You want to add active matter to it? Give me 5 years and a salary, and we'll talk.
Yeah. Great. Yeah. Well, congratulations, you got that done.
Yeah, it's good.
All right. Looks like Mehul's joined us. Hello, Mehul. How are you? Yeah, so... oh, hello.
Hi, Bradley.
Hi,
everyone. Yeah, I'm doing good. I actually saw this message of course material which you have mentioned. So, yeah, this looks interesting. So, is there a course from 2019 which teaches computational biology, something like that, in respect to DevoWorm? I actually was.
Yeah, I don't think we're doing anything with in parallel with that. So it's just kind of that thing that was posted. Morgan's really good at finding events and things like that. So.
Okay, sure. I thought it came from a previous meeting which I missed. So.
Oh no.
No, sorry. What was the question?
Oh, I guess you posted some things on the conference and things like that, and he wanted to know if DevoWorm was a part of that. And I said, not really, no.
Okay. Sorry, in like calls for involvement?
Yeah, something like that.
Oh yeah, I again, these are things that I aspire to go to, but have no direct involvement.
That's okay.
But I try to check to see if there's potentially a remote track or any will they film things or things like that.
Oh yeah, definitely. These Zoom meetings are always good to catch up on.
And
yeah. So yeah, we're not doing anything in conjunction with that, but actually, we should try to do more of that. I don't know. We can get involved somehow, but we'll see.
Which one was interesting, Mehul?
So you are talking about specific modules. So I was actually looking at the website only. I was looking at some hyperlinks. I didn't see specific module.
Gotcha.
Yeah. Okay. Yeah. Yeah. So we should talk about that today. We're going to talk about the Combine Harmony workshop. That'll be interesting in and of itself. And one of the people who was at Combine Harmony apparently is James Glazier. We just talked about him with respect to CompuCell 3D. So CompuCell 3D, I think it would be a great thing to kind of get to use in the group, but it's hard to get to, to sort of get it up and running for specific models. And I actually visited where they were at Indiana University back in 2015. So it was a pretty early days for DevoWorm. And I went there and, was trying to figure out how to use CompuCell 3D to model something. And they have stock models in CompuCell 3D. So I should say that CompuCell 3D is this platform. It's a agent-based modeling platform where they use cellular POTS modeling, which is a physics technique to model cells and cell populations, sheets of cells, things like that. So a lot of the things that we talk about in development, like epithelial sheets or embryos or whatever, those are the kinds of things you can model. Cells that are in sort of connected to one another in a configuration of some sort. And what you can do with cellular POTS models is you can introduce physics simulations. You can specify the shape of the cells and their interactions, and it could be like physical interactions or chemical interactions or whatever. So they actually have done a lot of stuff, interesting things with different types of templates. One of them being I think they've done a chick embryo, and I'm not sure what their state of the art is in terms of embryogenesis that they've looked at. They've done a lot of things with human organs and cancer research. So they've done a lot of things with tumors, tumor invasion. You have things like the liver, modeling the different tissues and cell types in the liver. And then, you can actually use real world data to train the model. And what I mean by train the model is not like you do with machine learning, which is to, train a network. It's more like you specify things, you parameterize things with real world data. So, if I have some microscopy data that tells me about, what the cells look like, or, maybe some videos of how the cells migrate across a space, you can use those kind of data to sort of parameterize your model. And that's basically, that's the best we can ask for in simulations is to get that parameterization. So yeah, that's something that's kind of in the midst of the past. So I don't know, I've never found anyone has really been able to get it to get CompuCell 3D up and running and really kind of attach it to a project. So, I don't think it's just something you can tack onto another project. It's something you really need to spend some time learning and, kind of finding the right model system and the right model. So, we talk about all sorts of different embryo genetic systems here in the group. We're interested in salamanders, which might be a good candidate for this. But there may be other systems as well that you could use. So yeah, so that's CompuCell 3D. But actually, there's this Combine Harmony event that happened about 2 weeks ago, and it sounds like a weird name, and I don't know. I think it was in the UK, and it had a bunch of people who are doing modeling, doing computational biology, and so I thought we should take a look at what they have and what they're up to. So this is actually, I think that the whole thing is the Combine, Computational Modeling in Biology Network. So this is an acronym for this group. Computational Biology or Computational Modeling in Biology Network, Combine. as an initiative to coordinate the development of various community standards and formats in systems biology. So if you know anything about INCF, that's what INCF does for computational neuroscience. So Morgan is asking about that earlier. What are the connections between computational neuroscience and then computational systems biology? And I think one of the areas where they kind of come together is in this community standards and formats area. So you'll have a lot of data collection, and a lot of groups are doing data collection, and then they want to share the data. Sometimes that's by mandate of the funding agencies. Sometimes it's just because that's what you do in a community, is you share data and you make it available to other groups. And so you can do that, but you have to be able to format it in the right way so that other people can access it. They can understand what's going on in the data, what the variables mean, and etc. And so these are, there's a lot of formatting involved. So having formats that are both accessible to other people and formats that don't make the file obsolete in 10 years, because we want to use these data for the long term. We want to have a long view of data sharing.
And Bradley?
Yeah.
Am I right in that Patrick Gleason is a contributor to OpenWorm?
Padraig Gleason, you mean?
Or is it Padraig?
Padraig, yeah.
Okay. I think I see his name in GitHub on OpenWorm.
Yeah, so he is actually an OpenWorm contributor. Yeah, and he's here too.
Okay.
So that's good. Yeah. Yeah. Okay. So the formats, and then the community standards, which are, of course, we all want to do things in a similar way because we don't want people pursuing wildly divergent methods, and then people don't know what to expect. So, like, if 10 groups are generating data, they should all use generally accepted standards so that we can, if I'm the end user of those 10 data sets, I can put them together pretty quickly. I don't have to question whether I got, whether I extract all the data out of the format or not, and I can really kind of merge the data sets easily. And so that's what INCF does as well. So yeah, you mentioned that Padraig Gleason is on one of the local organizers. Yeah, he's in OpenWorm. He's also an INCF person. So they do a lot of things with standards, and a lot of people in OpenWorm. OpenWorm kind of comes out of that sort of tradition of data sharing. So INCF is sort of populated by people who are OpenWorm adjacent. So like, for example, Steve Larson's doctoral advisor, Marianne Martone, she's in INCF. She's a member of INCF. Like a lot of neuroinformatics type people. And that's really been a core thing of OpenWorm. Really kind of, one of the sort of core things in OpenWorm that allow you to do the things that you want to do in OpenWorm are from this sort of thing. So, yeah, Harmony is a CodeFest type meeting with a focus on development of the standards, interoperability, and infrastructure. So this is, again, this sort of data sharing, big data sort of thing. There are generally not too many general discussions or oral presentations during Harmony. Instead, the time is devoted to allowing hands-on hacking and interaction between people, focused on practical development of software standards. So this was held at University College London earlier this month. If this is the schedule. We have, we got to go to the right day. This is Monday, Tuesday. So we have NeuroML. We have a breakout book for each one, so I can put this link in the Slack, and you can check this out on your own. But there's NeuroML, which is a hackathon for neuron and network models. Annotating systems biology markup language models with SBGN, which is gene regulatory networks or gene networks. So it's a systems biology modeling of that. there's demo of Simmune Monitor and Analyzer. Then on Wednesday, they had VCell, which is another modeling platform, exploring Copassi from JavaScript and collaborative opportunities for Combine. We had the PTAB breakouts. So there's a lot of stuff here. OpenVT, which is a multicellular simulation. And then Thursday we have sort of the end of the conference. There's this one, continuing work towards reproducible stochastic biological simulation. So you can see there's a lot of stuff here that's in that area. Let's see. There's the abstract book. I didn't want to get into that too much, but I noticed some of the people here, they have power outlet instructions for 220 volt. So if you're coming from the US, you know kind of what to do. So I noticed there are a number of people here. I didn't get I don't know where I found the list of participants. Oh, here's people. I don't know if this is all the people attending, but these are the coordinators. So there are a number of people who participated in this. We're doing the systems biology stuff, and then systems neuroscience, and all the adjacent areas. I know that James Glazier was a part of this, and of course Porrig Gleason. And another person who was involved in this was this person here, TJ Sego, who's from the University of Florida. And this is a paper that they wrote. I think this came out in 23. And I think Dick sent me this paper. And then I think Morgan also expressed interest in it. So this is TJ Sego and James Glazier on this. And so this is, kind of a group effort. It's an approach called Tissue Forge, interactive biological and biophysics simulation environment. So this is a specific type of thing that they're developing for looking at particle physics models and chemistry models and biology models to model tissues at the tissue level. So Tissue Forge is an open source interactive environment with particle-based physics, chemistry, and biology modeling and simulation. It allows users to create, simulate, and explore models and virtual experiments. So this is not just modeling, it's virtual experiments based on soft condensed matter physics at multiple scales. So you have different scales of soft matter physics or condensed matter physics going on in a cell or in a tissue. So you have cells interacting, you have things interacting, molecules interacting in cells and between cells. You have then tissues that interact and so forth. So you go from the molecular to the multicellular. And one of the benefits of Tissue Forge is putting this in a simple, consistent interface. So, people have tried to approach this multi-scale modeling problem by taking different types of models and combining them. So like you might model things like in a molecular simulation environment, and then you might model something else in a cellular modeling environment. And you might model tissues in a different environment. Like if we want to look at tensegrity, like Susan, we might use something like CompuCell, but CompuCell doesn't give us that sort of any of the biological stuff that we might want to model as well. So it's there's
no material in it that says this is a microtubule.
Yeah,
it doesn't have that, and you enter it in yourself, and it's still not right because microtubules can be pulled apart, and the cell still functions. It just grows microtubules in between itself, or something shifts. So, and CompuCell calls that an error.
Yeah.
And it really isn't an error. And the same with microfilaments. If they're compressed, you're not supposed to be able to push a rope, but in a cell it doesn't care. It just tightens that up itself. Like it says, oh, that's slack, and it there's a mechanism it has for pulling that taut again. Like there's several mechanisms. So
yeah.
I can't remember the name of the molecule, but yeah.
Yeah.
I do a talk on it actually.
Oh, yeah.
How does it do this? Well, it's not just pushing a rope in a cell. It actually has mechanisms for taking care of that.
Yeah.
And including myosin motors and anyways.
Yeah.
So CompuCell is a very crude method for looking at cells.
Right. Yeah.
So it always thinks it's a bridge.
Yeah. So yeah, one of the things about this is you can, of course, you can monitor the simulation and change things in real time. But then there's this growing library of built-in model components, along with support for user-specified models during the development and application of custom agent-based models. So this is where you have these components that aren't just like physical or aren't just biological. It allows you to really kind of span that gap. In the subcellular, cellular, and multicellular modeling domain, most spatiotemporal agent-based biological simulation tools only support one cellular dynamic simulation methodology and focus on a particular problem domain with a particular length scale. So this is where we have, we have these modeling platforms that generally focus on one type of process or one type of problem or one type of scale. So they kind of bring up their own, work, CompuCell 3D, and then they also talk about Morpheus. So for example, in CompuCell 3D and Morpheus, implement cell model objects using the cellular POTS model. So Morpheus and CompuCell 3D use either the cellular POTS model or the Glaser-Grainer-Hageweg formalism, which is the GGH formalism. And so this only supports Eulerian lattice-based models, while others like PhysiCell or CHAST, which is an acronym, support modeling cells with Lagrangian lattice-free particle-based center models as simple point-like cell particles. So you can see that you have these two different sort of approaches. You have this sort of physical model using sort of physics principles, and then this other one which is which is kind of a Eulerian lattice-based thing, and then you have PhysiCell or CHAST, which supports a Lagrangian lattice-free particle-based center model. So you're just using different types of approaches, Eulerian or Lagrangian, and you make a bunch of assumptions going into the model. So if your problem isn't really suited for those assumptions, then you're not going to get kind of the thing that you want. It's kind of like with Susan's situation, where she's trying to use a program that would, it's really the only kind of program that can model tensegrities, but it's really a mechanical modeling tool. So it doesn't give you that support for biology. And in this case, you're thinking about how you want to sort of the perspective you take on how the biology works. So for example, you could use a particle model, you could use sort of a lattice-based model. And so you have to make those kind of decisions. And, depending on the system, it may one approach may be appropriate or the other. The second version can be extended to include subcellular detail using the subcellular element model, which could support modeling the spatial complexity of cell shape, cytoskeleton, and extracellular matrix. Now, extending the CPM-GGH, which is this first one, the cellular POTS model, Glaser-Grainer-Hageweg formalism, to include cellular compartments, allows representation of subcellular components like the nucleus, critical molecular species or regions with specific properties, but does not support specific representation of macromolecular machinery. So this is, again, we have these sort of subcellular components we can model, but it doesn't really help you too much, with other types of things going on in the cell, macromolecular phenomena. Typically, modelers who are interested in subcellular and cellular detail must use and adapt general-purpose molecular dynamics simulation tools such as LAMMPS, HUMD Blue, NAMD, or GROMACS. So these different molecular dynamic simulations that people use. For example, Shafi customized LAMPS to model cells as cluster of particles to simulate spheroid fusion during spheroid-dependent bioprinting. So you can use these kind of molecular dynamic simulations to do a lot of things, sort of particle-based problems. So, again, it depends on the problem you're trying to approach in your biological modeling. If it's something that's amenable to this sort of particle simulation or these molecular dynamic simulations, then you're unlocked. There are a lot of nice models. If that's not exactly what you're doing, then you have a problem. Most molecular dynamic simulation tools are designed to parse and execute models that are theoretically well-defined, and molecular dynamic simulation specifications and engines tend to be well optimized for computational performance. Most assume a fixed number of objects within a model and do not support runtime object creation, destruction, or modification. So this is something, where you have sort of a limited degree of expressivity of your code. That means that you can only do a finite, number of things. You can't do a lot of operations that you might be able to do if you just simply coded it in Python or C++, directly and use like object-oriented programming to do some of these things. You can build things at runtime, and you can't really build things at runtime, and you can't really modify objects, say. So that's a problem for a lot of these tools. In molecular dynamic simulations, they come out of, very limited set of use cases where you want to, say, model a bunch of molecules bouncing around in a container or something. And they're very good at that. They're very optimized for that. But they don't have a lot of the details that we need for general purpose modeling. So that's basically...
do they include gravity in their models? That's what
for these kind of models, I think they do, but I don't know how they put them in. I think what they're talking about here is just kind of like the basic sort of approach to the problem. So, like, can you represent your problem on a lattice? Would you put, like, is it a bunch of cells interacting on a sheet? Or is it like, something where you have molecules bouncing around or moving freely that aren't on a lattice? So you have the physics. The physics are kind of implied in the model, but the idea is that you're trying to specialize it for specific types of things. I mean, there's some basic physics modeling in this, but yeah, I mean, again, with molecular dynamic simulations, I don't think you can get very sophisticated with that. Like, I mean, like you're...
I asked someone one time if they had included gravity in their molecular model, and they said, no, gravity doesn't have anything to do with it. And I'm going, yeah, there's no convection.
Yeah.
and microgravity, you have to include that if you're going to look at gravity as an input. Anyways.
Right. Well, yeah, that I don't know. I mean, that very well could be that they don't have, like, they haven't specified a lot of those interactions, but I'm not sure, like, if you use the package, I'm not sure how specific you can make any one model. So they'll have, oh, go ahead.
You might be able to change the physics so that it includes it.
Yeah. But, there's also this issue of heterogeneity. So if you, say, go across your model, what is the specific gravity? What is the specific gravity doing, and so forth? How is it represented in the model? And does it represent everything equally? So you can have different convective forces as well, and that might be heterogeneous across the simulation. So there are a lot of things going on there. That I think the point here, though, is that a lot of the papers or a lot of the models that have come before this are sort of, not really, they're not even really standardized in the same way that you might expect a modeling package to be. They're specialized for specific problems, but if you go outside of that domain, then, that's your problem. And then, they make a lot of assumptions that a lot of times people, have to really kind of think deeply about to get, their problem to match what the software outputs. So, it's a lot like mathematical modeling, or like, thinking about like how to represent some biological thing, right? We want to represent an epithelial sheet, for example. How would we want to do that? Would we want to use an Eulerian approach or Lagrangian approach, or some other kind of approach, or maybe something that's, a minimal physics model, a minimal model? There are a lot of options there, but the point is that you want your model to be reproducible. You want it to be sort of interpretable. In other words, you want to know how it's operating, and you want to know if it's well suited to your problem. So, and of course, if you stack models on top of one another, they have to be interoperable, because it doesn't make any sense to use like 3 different tools for 3 different scales and then say, I'm attacking a multi-scale problem, and you can't fit them together very easily. So these are all problems. That's how they kind of motivate TissueForge. So this paper presents TissueForge, an open source real-time modeling and simulation environment for interactive biological and biophysics modeling applications. TissueForge is designed to address many of the aforementioned issues and challenges. It enables agent-based spatiotemporal computational modeling at scales from the molecular to the multicellular. Is designed for ease of use by modelers, research groups, and collaborative scientific communities. Here we describe the philosophy, mathematical formalism, and basic features that demonstrates usefulness across multiple disciplines in the physical and life sciences. We present representative examples of advanced features at a variety of target scales. So let me go down. So they have what they call open science support, which is this sort of aspect of data sharing and, model, sort of modeling standards. You have a number of computational benefits like interactive and batch execution, which allows you to sort of develop your models and run them and modify them in real time. So they introduced the concepts here. So for Newtonian dynamics, the position of ith particle in R is updated according to its acceleration, which is proportional to its mass and total force exerted on it. And for Langevin overdamp dynamics, m sub I is the drag coefficient, and particle velocity is proportional to the total force. So we have force calculations here, Susan. This is sort of what they're working from in terms of the kinds of physics that are represented in this model. Then they also support 3 broad classes of force-generating interaction. There's the implicit force, which is the force due to implicit interactions between the ith and jth particles. So if you have 2 particles, they're interacting, that's the implicit interaction force. Then you have a bonded force, which is the force due to bonded interactions between the ith and jth particles. And then the explicit force, which is acting on the ith particle. So you have implicit interactions between I and j, bonded interactions between I and j, and then an explicit force acting on I. So that's how they do that. And then the implicit interactions result automatically from interaction potentials between pairs of particles of given types. Bonded interactions act between specific pairs of individual particles. And then explicit forces act on particles through explicitly defined force descriptions and do not necessarily represent interparticle interactions. So, for example, gravity, internal noise, system thermal equilibrium. So this is, again, the question you asked. This is where you have these explicit forces that you can represent. You're not representing them well, I guess you could represent gravity specifically, but you want to turn things on and off depending on what you want. So I think that's why they leave that open. So sometimes internal noise in some systems, that's really important. System thermal equilibrium sometimes is really important. Sometimes gravity's, you might want to turn gravity off or turn it on depending on what you're doing. TissueForge provides built-in force and potential-based definitions and permits applying an unlimited number of executable force and potential objects to individual particles and particle types. So again, you can plug these in.
If you're going to subject your cells to centrifuging,
Yeah.
you need to include gravity.
Yeah, well,
that's a force. I mean,
sure,
it's
yeah.
that. Like, you can certainly wreck cells by centrifuging. You do it deliberately. But sometimes people separate cells by using centrifuge, and I still think that has an effect on cell type when you're all set and done with the centrifuging.
Yeah. And then, of course, if you're in a cell culture environment, you also have substrate. So if you're on a certain substrate, you can affect the fate of cells, or at least the way they interact. And so, but all these things, I mean, you can model them in different ways using different explicit forces. That's the idea. And I don't know, you could argue that it's not enough, but, that's what we do with modeling. Sometimes we're just dealing with like throwing in a generic thing and then saying it represents something. So this is a diagram here, figure 1, where they talk about the different modeling features here. So, in A, we have 5 superimposed snapshots of a double pendulum implemented in TissueForge. So they have, double pendulum is where you have a double jointed rod that comes down and swings on like a center of gravity, around a center of gravity on this thing that's sort of stable. And then you have these 2 things that move back and forth. So it's like a pendulum, but there are 2 independent parts to it with a joint in the middle. And so you can have a large number of configurations that result from that. That's what they're doing here. So they're able to demonstrate that you can model kind of like these interactions between linked particles. And so that's kind of what they're doing there. Yeah.
A double pendulum like that is actually a chaotic system.
Yes, it is.
Yeah, interesting.
So the dynamics are going to be chaotic, if you plot them out at the different joints. And so B is 4 TissueForge clusters. So these are the particles that are different colors, and they're clustered, representing biological cells, each consisting of 10 particles, whose color demonstrates cluster membership. Potentials describe particle interactions by whether they are in the same cluster, intracellular, or different clusters, intercellular. So a blue cell and a yellow cell have a relationship. Those can be an I and J, but they're in different clusters, so you want to treat those differently. C is TissueForge simulation of chemical flux during fluid droplet collision. So this is where you have blue and red, these 2 particles, I and J. They kind of come together and they splat, and they're kind of overlapping here. And then those parts are kind of coming apart. So they can model sort of particles that come together and they form like patterns. They sort of form droplets from the drops and splatter out. And you can see sort of this simulation of this process here. So this is chemical flux during fluid droplet collision. Each particle represents a portion of fluid that carries an amount of a diffusive chemical, the amount of which varies from 0, which is blue, to 1, which is red. So you have 0 and 1, and they collide, and then they create all these intermediate sort of patterns and colors and amounts. When 2 droplets carrying different initial chemical amounts collide, resulting droplets tend towards homogeneous chemical distributions. So you can see that here. So they can do a lot of interesting things with the physics here. Definitely more than you can do with CompuCell 3D. So, this is something that we were interested in with, like, at the molecular scale, but also, maybe relevant to the cellular scale as well. So they kind of go through some of the more of the math here in terms of what you're kind of the different functions that you're using within the different types of interactions. Since TissueForge enables the implementation and execution of models at different length scales, particles in a simulation may represent objects with a wide variety of possible behaviors. A particle could be atomic and subject to energy-conserving implicit interactions. So these different types of potentials that you can use here, as in classical molecular dynamics. Particles can also represent portions of material that constitute larger objects. So, for example, a portion of cytoplasm, and can carry quantities of materials within them. And that's an example of that is the convection of a solute chemical in a portion of fluid. Tissue Forge provides built-in features to enable particle-based modeling and simulation of fluid flow based on transport dissipative particle dynamics, or TDPD, and smooth particle hydrodynamics, including a predefined TDPD potential that can be applied when describing the interactions of a simulation. And they have an equation here. And so, there are all sorts of ways we can treat these kinds of dynamics. We can also do things allowing particles to carry embedded quantities, or treating particles as constituents of larger objects. So that's a cluster for embedded quantities, like we saw with the droplet splattering. We can actually attach to each particle a vector of states that can evolve during a simulation. So you see that you can actually have it within a particle. You can have a lot of different states, and then you can actually break those apart as you need to. So there are a lot of ways you can represent this mathematically in terms of code, and so that's really nice, some of the stuff that they have in here. And you can represent flux. You can represent different types of like chemical species transport. And then they kind of get into the code here where, we have these, we're working actually on modeling and simulation specification using classes, objects, and functions. So we're using basically object-oriented programming. And so, this is a very powerful technique where you can assign different sort of conceptual categories to objects, and then break that object down into instances and other things. So, we can build a model of, classes, objects, instances, things like that, and that can be related to a real-world system. So if we want to model a cell, we might have different classes of things in the cell. We might have different objects. Those objects are going to have instances, and we can actually just write this in code to simulate this. So this is, something you can do in either C or Python. And they show some examples here where this is in Python where you're importing Tissue Forge. So, they have the package available. You just import it into Python, and then you start running this object-oriented, this sort of framework. So specifying a particle type with a particular radius, you're using a class from the toolbox. And so you can, initialize the simulation, retrieve an executable particle type, and create particles all in Python. And it's not that much code, but they give you these examples so that you can do things in code. And then this shows you sort of an example of what a simulation will look like. And I don't know how they're visualizing this, but basically you have these two particles. You've set up a simple oscillator interacting via harmonic potential. So, we're putting all that stuff that we've talked about together to do this. And then, of course, we have the, this is at the, I guess this is a, let's see. This is actually using the Python API to specify an interactive simulation of a convection of a species near a species sink in Python. So this is actually where you're using something in a Python interpreter, and then you can also use this in a Jupyter notebook. So Jupyter notebooks are very lightweight compilers. They allow you to execute things in kernels, and we can put them on GitHub. And you can see that the quality of the simulation is basically equivalent. So if I can build something in Python interpreter, and I can put it online in GitHub, and people can work on this, kind of interactively. So this is very powerful stuff. It allows you to do a lot of things, not just in terms of simulation, but also in terms of implementing it in code. and doing it in a good way that we can then share with our community and play with the parameters, and it's pretty transparent. Okay, so I had a comment.
Well, thank you for that reference.
Oh,
I'll
yeah.
write it up
Yeah.
in my presentation because it has a nice summary of a bunch of the simulation packages. So, and adds a new one.
Yeah. So I guess, yeah.
All of that's very nice.
Yeah. So yeah, I guess the lesson here is that, we have these very opaque specialized tools, and we're kind of bringing them all together into this very interactive, easy to use package, and that's why they're kind of pushing this. And so, we should maybe look into this as a way to model some questions or problems that we have in the group. I'd be interested to see what people can do.
It does do convection. It just said that was a model using convection.
Yeah.
So you could do it where you have no convection or very little, and then a lot more convection than you get at Earth's gravity. And you could do simulations on the moon, and you could do simulations on Mars, and it could make it interesting that way.
Yeah.
By changing the convection.
Mm-hmm.
Convection rate.
Yeah. That's great.
Yeah, so there we should do a project.
Yeah, see what it can do. All right. Well, any other questions or comments before we go?
Yeah, that's a great package, though. Yeah, and great project to follow.
Yeah. I don't know where it is on GitHub, but is that?
It's okay. I'll look for it.
Okay, yeah.
I'm interested.
All right. Sounds great. All right. Well, thanks for attending, and yeah. All right. See you next week. Thank you.
All right.
Bye. Take care.
Bye.
Bye. So for those of you interested in TissueForge and the GitHub repository, here it is. It's github.com/tissue-forge. And this is what they have on their README here is they have a number of really interesting particle simulation examples. So this is kind of an example of everything we were talking about. We have collision, splattering, sorting into these groups. You can see all of it right here in this top image. And so they have a build status here for the binaries. They have a set of documentation examples. So, this is for the API and for the basic program and for Python API, C++ API, and so forth. They have a gallery here of specific things. These are animations of things we saw in the paper. So this is collision of droplets in the resulting splattering. This is where you have this cell sorting in a tissue by differential adhesion. This is where we have a vertex model, which is configured sort of a model of local vasculature that, you can build a model from components, and, you're using this mesh to build the model, and you're exporting the mesh, and you can actually, so this object file is generated in Blender, and you can use that as part of the model. Then you have these two-dimensional agent-based models. So this is a two-dimensional sheet of cells that it's a two-dimensional kind of like a cellular POTS model. This is cell proliferation and differentiation in the colonic crypt. So this is a specific organ where you have a bunch of cells and they have these different states. So they're doing this in a very similar manner to the molecular dynamics or the molecular simulation, but they're using not molecular dynamics nor cellular POTS modeling. They're using this approach. Then you have this simulation of single cell migration over an extracellular matrix across the fibrous. So this is actually a nice example of like a cytoskeletal structure and cell migration within that structure. And so, this is something I think that might be useful to some of the modeling Susan's doing or some of the modeling of within the cell that we might want to do in our group. Okay, one thing I'd like to talk about with respect to combine harmony is that combine, of course, is a set of standards that are sort of where they're going with that. So they actually have this nice resource. They have these Jupyter notebooks that show how to create simple models from each of the combine standards. So it's one thing to have standards written down in a place. It's another thing to have like a notebook that shows you how to execute the standards and how to put your data into the standards. So there are a number of standards here. There's CellML, there's NeuroML, there's OMEX, there's SBGN, there's SBML, there's SBOL Systems Biology Open Language, and there's SATML. So this is a simulation in SBML. So each of these have their own GitHub repository. They have actually a binder server. So this is really interesting. This is really good. And then they have this main examples repo here, which is combine.org on GitHub. So this is combine.org. These are their notebooks. And if you go to their organizational front here, this is the Computational and Biology Network, and these are their repositories here. So they're doing a lot of stuff with, educating people and putting out the specifications and other things. So another thing I wanted to talk about is this DevoWormML repository. So this is, if you go to github.com/DevoWorm, there's this repository, DevoWorm-ML. And so this is this DevoWormML class that I talked about in the 10th anniversary talk. Is something we did in the fall of 2019. And this is the last time it was offered. And, this was separate from our regular meetings. So this was an hour-long format where we would go over pretty much the 16-week fall term of a standard university and, going through different topics from week to week. And so, this was something that was meant to be educational for biologists who wanted to learn about machine learning, but also for machine learning people who wanted to learn how to apply their models to biology. So we have this repository, and you're welcome to issue pull requests to it. You're welcome to go through the materials. You're welcome to, so we had, for example, we had these 5-minute reviews that people contributed to. We had current topics, which of course, since this was offered in 2019, you can contribute to as well with new content. And hopefully at some point we can do this again, because this is something I think that needs to be updated. And as evidenced by conversations in the Slack and in the meetings, there's some interest in doing this again. So we have this stub on our website. This is DevoWorm.weebly.com. And it kind of goes through the syllabus here. So we had a 16-week schedule. It starts with sort of this informational first meeting where I introduced this knowledge map of machine learning. And, there are a number of different things that we talked about in that meeting. The second meeting was on a project that we've done called Digital Basilaria, which is modeling a very small organism. It's a type of diatom that lives in colonies, that they have this really interesting set of movement patterns. The third meeting was on something called Open DevoCell, which was really the precursor to DevoLearn. So this was pre-DevoLearn. In fact, DevoLearn came out of this effort. So this is an interesting kind of historical connection within the group. And Open DevoCell is really, about applying RNNs and CNNs to microscopy data. So this was the main thing about the third meeting. This is the hype cycle for artificial intelligence for 2019. This was, I think, part of the first session, but it's interesting to look at this both in terms of where these technologies are on this hype curve and historically. What does the hype curve look like for 2024? And I'm not going to get into this curve, but a hype curve is basically something that the Gartner consulting firm puts out on different industries. And the idea is that, there are these things that sort of get these innovations that occur, these models that get released, and they have this, there's sort of this peak of inflated expectations where everyone thinks that it's going to solve all the problems in the world. And then there's this trough of disillusionment where people say, well, this model is pretty much flawed, maybe fatally so. And then there's this slope of enlightenment and this plateau of productivity. And so the reason we have this in here is because a lot of the techniques that we talk about, a lot of the models follow this kind of hype curve. So you go from a model being released to something that everyone adores and they think it's going to be applied to every problem in the world. And then when it invariably does not do that, then people kind of go overboard in saying that it's not worth our time. And then eventually we figure out the best kinds of models for different problems and we kind of adapt those and use those. And so you saw in the meeting with some of the models of cellular physics that we have different models that people use, and they don't necessarily go through this hype cycle because it's not, a commercial endeavor, it's an academic endeavor. But I would say that for a lot of biological modeling, we could build a similar curve, sort of an expectation cycle. And so that's another aspect of this course that might be of interest to people if you're interested in technology. Then we had a very important fourth meeting lecture on input data, and this is something that I presented, and this is a very important thing for a lot of modeling in general. So when you have a model, you want to have good input data, especially for machine learning, because you need to be able to train your model, you need to have it reflect the real world, but you also want it to reflect a lot of the edge cases and out of distribution aspects as well. So biology is rife with a lot of behaviors that are hard to actually, classify very neatly. And even when you have a lot of data, you won't observe some modes of behavior. So this is, talking about input data. This is something I'm probably going to update, but, if you're interested in machine learning, this is an essential topic. But it's also, as we saw in the, biological modeling space, this is also something we need to think about when setting up our models or initializing our models. So in machine learning, we don't worry about this as much, but maybe we should, because machine learning, we need to have a model that captures the right assumptions about the system. But we also need to have, in biological modeling, we need to pick the right approach, the right assumptions to match what we want to model. We can't model physics correctly if we don't have the right sort of framework. So this is a very important topic. And if people are interested in contributing in any of these areas, I suggest, maybe issuing a pull request in the repository, and we can maybe put something together, some sort of parallel activities. We could put them on the syllabus. So the fifth meeting was about pre-trained models for biology. And this is, again, kind of where we were trying to go with this in part. This is the thing that actually led to DevoWorm. So we thought about, what's the best way to approach biological modeling and using machine learning. But one of the ways to do that is to pre-train a model. So what you do is you take a bunch of data and you put it into a model and you train it, and then you deploy it in different contexts. So for example, if I wanted to look at C. elegans development, I could train it on a lot of the labels that we know, a lot of the basic data, and then we can then take that model as it's pre-trained and apply it to different data sets in C. elegans. But we can also apply it to other organisms as well. And while that has limited utility, the pre-training aspect, it does give you some information that you can then use to improve the performance. So pre-trained models, this is something that we wrote a blog post on, and we posted it, and then we ended up pursuing that for the next year's Google Summer of Code project, and we ended up building DevoWorm from that. So sometimes these topics can lead to big things. This is, a random kind of thing that was put in here. This is actually in cognitive computational neuroscience. So interestingly enough, Morgan in the main meeting asked the question about the connection between computational neuroscience and computational systems biology. And this is actually maybe a connection here. So this is from this paper, Cognitive Computational Neuroscience. It can be found on the archive. And this is the figure that this is sort of their idea of modeling. And this actually applies to not just computational neuroscience models, but modeling more generally. So on the x-axis, we have what we call biological fidelity, which is the sort of the truthfulness of the model or the realism of the model. And, we can go from like the Hodgkin and Huxley model, which is a very basic minimal model, mathematical model of conduction and neurons. We can go then to a multi-compartment single neuron model where the model of neuron is a bunch of processes that are in different compartments. This is much like the stuff we were talking about with reaction diffusion modeling. And then the Blue Brain project, which is actually like modeling a brain with neurons, with interactions between the neurons and so forth. And so you can see on this side we have model complexity. So the smaller circles are sort of the minimal models. The big circles are the sort of the hyper-realistic models. And on the y-axis we have cognitive fidelity, meaning that we have, in this case, things that incorporate cognition more or less. And so we can see that we go from the Gabor wavelet V1 model, which is a minimal model, mathematical description of V1, which is the visual area in primates. We have these DNN models or DCNN models. These are deep neural networks, and they're kind of at a mid-range of cognitive fidelity. And then we have cognitive models, which are a little bit more complicated. And these are just kind of subjective sort of rankings. I think cognitive models may be a little bit higher on this axis, but the point being is that you have this cognitive fidelity, you have biological fidelity, and you have this model complexity, and so we can plot all these different types of models out in this way. And then we have these processes. So a lot of cognitive models are modeling two different things. They're modeling top-down processes and bottom-up processes. So top-down processes are where we have mental models that kind of inform what cellular models are doing. So we might have labels, we might have concepts, and we map them to cells and how they're kind of interacting in a network. Bottom up, on the other hand, is where we have the activity of a network or of constituents, and they inform the labels and they inform emergent phenomena. So we have this top-down and bottom-up aspect. And, models can incorporate both, but generally they're in one of these two streams. And then you have what we, want to sort of do, which is model the brain, model cognition. And we do this computationally. And so we reach this gray circle at the end. So that's kind of one of the aspects of, sort of the meta aspects of machine learning that you'll learn in the course. Then in the 6th meeting, we had a TensorFlow tutorial. So this is a little bit dated because we don't have a scikit-learn tutorial, but we also, TensorFlow has been improved since this is given, so we probably need a new tutorial on that, but I think, the basics of this are all basically going to be correct. Just setting up an environment and running a model in it, and that's kind of what we want to do. We were involved in Hacktoberfest that year, so we wanted to solicit some contributions to Hacktoberfest, and we solicited a few. If you look in the repository, we have our 5-minute reviews, and these are reviews of different topics that you can, kind of summarize and present to the group, and then you become a contributor. In the 7th meeting, we had a general discussion. This was kind of strategizing about this blog post, which is published on the Node, which is a blog for developmental biology, pre-training models for biology. So this is something that led to our GSOC project in 2020. We also, in the 8th meeting, talked about computational pareidolia, which is this phenomena where models will see patterns in random sets of data. So, if you think about, like, if people see Elvis Presley on a piece of toast, or the face of, like, some famous celebrity on the surface of a tortilla or something, and it's just like the toast marks or the burn marks on the food, but you see this pattern, and so there's this phenomenon, and it's not necessarily model hallucination, which is a very popular topic, but it's very specific to like image data, where you'll see patterns in in the image data that look like something else, and the computational model picks up on that and misclassifies the thing. So misclassify some feature in the data set as something else. We want to avoid that. It's it's sort of analogous to hallucination, but it's very important. It's a very interesting topic too. So we cover topics like that that are kind of outside the mainstream of a traditional curriculum, but are very relevant to biological modeling. Then we have developmental GANs and GANs in medical imaging. So this is, we had someone present on this where they talked about medical imaging and using GANs and developmental GANs. And we use GANs in DevoWorm as a generative model of embryos. And it's a very interesting thing that's been underexplored. We do have that capability in the software, but we haven't really done a lot with it. We can do things with developmental GANs as well, and GANs are kind of, have kind of gotten lost in the shuffle of machine learning and deep learning, but they're very useful. There's a lot of gas left in that tank. They're actually not that old of a method, but they really do some interesting things, especially adversarial GANs. So, I guess they're all adversarial. So this is something that we could follow up on as a project. Medical imaging, of course, is just taking modeling, finding features in biological image data. But developmental GANs are more about like modeling this sort of emergent process that we define as embryogenetics. So we have different slides for that. All the slides and videos are available online, and I'll show you in a minute where that is on our YouTube channel. This is our blog post. We had another general discussion in the 10th meeting. In the 11th meeting, we had game theory and machine learning and development. This is actually draws from the GANs literature, because in GANs we're kind of implicitly discussing game theory and some of the aspects of game theoretic approaches. And so this is something that I talked about in a lecture. And we not only can do this in machine learning, but there are applications of game theory to development as well. So there's this intersection of game theory in both of these areas. Then in the 12th meeting, we talked about reinforcement learning. In the 13th meeting, we talked about biological metaphors in machine learning, reinforcement learning, and cybernetics. So we brought cybernetics in terms of biological metaphors. So biological metaphors are very important when thinking about larger systems at the larger system scale, because we can model things at sort of the feature level. We can model things in terms of labels, but in development we're dealing with these emergent features that kind of come out of a bunch of cells interacting. So as you saw with Tissue Forge, you have two droplets that splatter, and and their components end up all over this field that you're modeling. So there are emergent patterns that result from that splattering. That's a very simple action, and it has a lot of consequences. And it would be hard to identify the splatter. That whole process is a set of features. And so we need to use metaphors. And maybe, using metaphors, like the splatter is like something, and that something can be modeled using this method. It helps you do a lot of problem solving and finding the right model. But it also allows you to sort of model the process with sort of a general approach in mind, instead of just saying I'm modeling splattering. Splattering is also similar to some other thing that's happening in biology, and maybe we have this model or this technique that can sort of cross a bunch of areas. The 14th meeting talking about this technique called deep fovea, medical imaging, and Peter's rule. And these were these different topics, these 5-minute reviews, and kind of going over some topics, some techniques like deep fovea and Peter's rule, which is a specific type of thing in machine learning. And then in the 15th meeting, we talked about invariance and universal features. And again, this is a lot like the input data lecture, in that it's very much like, how do you model and what are you doing when you model? In this case, thinking about invariance, sometimes we're modeling systems that are invariant. So in other words, if I go to development, I can use techniques that maybe are used for medical imaging, or used on embryos from different species and use them in different, they're generalized in that way. But sometimes they're not. And sometimes features are very specific to a specific data set or a specific organism. So we want to think about invariance and universal features a bit differently in biological modeling and in applying machine learning and deep learning to biology. Because in a lot of that literature, in the deep learning and machine learning literature, we assume invariance, or we assume universal features across data sets. And we use benchmarks, but the assumption is that we can get features out of the data in much the same way all the time. And so what I'm saying here, we need to think about that and really kind of test that assumption. And then here we have this paper, Hydrodynamics of Embryo Cell Positioning, as an example of what's going on here. So, we covered papers as well as topics. So this is another paper kind of, excerpt, artificial neural architectures and a minimization function for the brain. And that was posed as a question because, can we use minimization to model neural architectures? Does it work on the principle of gradient descent? And so this is an example here in this paper, a deep learning framework for neuroscience, Nature Neuroscience. And, this is kind of answers this question. So you have this, it's theoretical, but you have these architectures and you consider, what is going on in this gradient descent architecture. So if you have this architecture, you have this space, it's a gradient descent space, you use an architecture and you end up with these different aspects of the space. So for example, when you have an objective function, the objective function can either get better or worse, and it's either climbing or descending this gradient. We also have different parts of the gradient that represent different synapses and how strong they are, so you can look at different learning rules, and it shapes where you are on the gradient. And then, of course, if you can get some sort of optimization, what does that mean in terms of the architecture, and how does that map to a brain? So this is all interesting stuff. Then the 16th meeting, we kind of summarized hierarchical temporal modeling in new directions. So this is where we're going in different directions other than just machine learning. Hierarchical temporal modeling is this approach developed by Jeff Hawkins and the Numenta group. So they're doing things modeling neocortex and the layers of neocortex. And so, we can use that as inspiration for models. And the Numenta model, actually, they don't just, put some sort of bio-inspired model into practice, but they're able to tackle a wide range of problems using it. So we consider some of these alternative models. I think we got a little bit into bio-inspired models, and neuromorphic models, but that's something that maybe we can update the course with. We also have Rethinking the Bias Variance Trade-off, which is a paper which is also important in looking at machine learning performance. You want to have a lot of variance in your data set without having bias. And so there's this famous trade-off that we discussed. And then there's skepticism of backpropagation in AI, which was a discussion on Quora. And this was from Jeff Hinton posted a question about backpropagation, or I don't know if it was Jeff Hinton, but Jeff Hinton was the sort of person who developed, or did a lot, well, I guess it was Jeff Hinton. So Jeff Hinton, poses this skeptical view of backpropagation being sort of this feedback between layers in a neural network, and whether it's something that is either observed in biology or really is something we can rely on. So this is some really interesting things, bringing things from different areas. We also have additional content which were kind of after the course, so a lot of things in 2020 and 2021 where we kind of did little lectures and different features on things that were outside the scope of the basic curriculum. So these were parts of the lab meetings that we do weekly. So we talked about transformers for biology, spatiotemporal feature extraction, something called torch streams, which was something that was developed by one of our GSOC students, which is interpretable deep learning. developmental data for model training, neural style transfer, ResNet voice recognition, diagram tracking using object detection. This is again that digital diagram project. Reconstructing continuous biological processes with deep learning, neurocellular automata, NCAs. This is differential morphogenesis, and deep learning for the life sciences. So again, all these are application domains. Some of them are maybe less relevant to biology than others, but I think we're really interested in any model that exists. We haven't talked about large language models at all, but this is something we talked about more recently in our group meetings. And so, I may actually add those segments, because these are just segments of the lab meeting. So, it it's queued up to that discussion. So I may add some things into this. But I'm looking for people, I'm looking to sort of update this curriculum, build maybe some new lectures, or, really something that's outside the scope of modern sort of pedagogy in this area. Because a lot of the educational opportunities one has is to learn about machine learning at a very technical level, learn maybe about biological modeling somewhere, and then you have to put them together. And what our group does is we put all this together immediately, and in some way that's unique, that you learn insights right in the class instead of having to go outside of the class. So as a finale here, I'd like to highlight DevoWormML lectures on our YouTube channel. So we have a playlist called DevoWormML. If you go to our YouTube channel, DevoWorm, and you look for DevoWormML, and under the playlists, you'll see that we have all these lectures from the course. And so these are, I think all of the week, well, some of the weeks are not on here, but most of the weeks are. We have the inaugural meeting, week 4, week 5, week 6, week 7, week 8, week 9, week 10, week 12, week 13, week 16. So if you're interested, check it out. Check out some of these video segments as well and some of these models. And these are just by our contributors and their discussions on the stuff. So it's not, the ultimate authority by any means, but it's really our sort of perspective on things. And so I look forward to people contributing to this, and thank you.